Greetings to one and all. I'll be presenting on gliomas, an experience based on molecular markers. I am Dr. Yookarin Khonglah, Associate Professor in NEIGRIHMS, Shillong. So when I was requested to make this talk, our study was done from 2017 to 2019, and it was in 2016 when the WHO CNS tumors classification incorporated molecular parameters with the histology to define tumor entities. And this was done in the hope to aid in the diagnosis and prognosis of gliomas. But we published this paper in 2020, and then came the new WHO classification 2021. So the obvious question for me was our paper really relevant now or not? So I will answer that question after you listen to what study we have done. So gliomas account for 45% of all intracranial tumors with an incidence of 4.6 per lakh persons per year. IDH mutation present in 70 to 80% of grade 2 and grade 3 astrocytomas, 80% of oligodendrogliomas, and 82% of secondary glioblastomas. It is very rare in primary glioblastomas, and it is absent in ependymomas. This sentence, please remember, because we have excluded these in our study. The clinical relevance of IDH mutation is that it confers a better prognosis and it renders gliomas more chemo and radio sensitive. The next molecular study which we did is the Atrx gene mutation, and this gene mutation leads to loss of expression of Atrx in the tumor cells. Atrx mutation is a specific marker for astrocytomas, but it is retained in oligodendrogliomas. P53 mutation is seen in 58 to 83% of astrocytomas. Secondary glioblastomas also show P53 mutation. Primary glioblastomas and oligodendrogliomas rarely show P53 mutation. So the objective of our study was to correlate and analyze the variability of expressions of mutant IDH1, Atrx, and P53 proteins in different types and grades of gliomas. And our secondary objective was to follow up and attempt a prognostication based on the molecular findings. The study was approved by the Research and Ethics Committee. We included the retrospective and prospective cases from 2013 to 19, and we took all the relevant clinical and radiological data. We recorded light microscopic findings and did the WHO histologic grading of tumors. We excluded cases of ependymoma and primary glioblastoma because they do not show mutant IDH. So immunohistochemistry for IDH1, Atrx, and P53 were done by the horseradish peroxidase method, and they were stained by the monoclonal antibodies listed here. And each immunohistochemistry slide was examined by 3 independent observers, and they were blinded to the clinical and the histologic data for objectivity's sake. So how to interpret these IHC stain? So IDH R132H clone was used, and if there is moderate to strong cytoplasmic positivity or a perinuclear positivity or a weak nuclear positivity, all these are taken as positive, and this is labeled as IDH mutant. Atrx, if the nuclear positivity is there, it means that it is not mutated and the staining is retained. Whereas if there is no nuclear staining, then we say that Atrx mutation is present because there is loss of nuclear staining. So, but we need an internal control in such a case, and we can look at the endothelial cells and lymphocytes which act as internal control for Atrx. P53 stains were graded as 0 when no cell stains nuclear positive. When less than 10 cells show nuclear positivity, that's individual cells, it is 1+. When there are clusters of cells showing nuclear positivity, that is 2+. When all tumor cells show nuclear positivity, that is taken as 3+. So in the results, we had a total of 53 cases with a male to female ratio of 1.3:1. The frontal lobe was the most common location, and headache was the most common symptom. The histologic diagnosis, we had 2 cases of pilocytic astrocytoma, 9 cases of diffuse astrocytoma, 10 cases of oligodendroglioma, 5 cases of anaplastic astrocytoma, 5 cases of anaplastic oligodendroglioma, 22 cases of glioblastoma. So in total, 53 cases. The WHO grading grade 1 were the 2 cases of pilocytic astrocytoma, 19 cases were grade 2, 10 cases were grade 3, and 22 cases were grade 4. So we look at the clinico-pathological parameters such as the age, the sex, the WHO grading, and the histologic lineage, and compared it with the IDH mutation. And we found that there is a correlation only when IDH mutation is correlated with the WHO grade. As you can see in this table, that 89% of low-grade gliomas are positive or they are IDH mutant cases. And again, we looked at the association of Atrx with various clinico-pathological parameters, and we found that the histological lineage was significant, statistically significant. And you can see in this table that all the cases of diffuse astrocytoma show Atrx loss, and all the cases of oligodendroglioma show Atrx retained. That is the significance. We also looked at the association of 2 of the molecular markers versus the WHO low grade and high grade. And we also looked at the association of 3 of those molecular markers with the grading of low and high grade. And we found that both were equally statistically significant. Out of the 53 patients, we had a follow-up of 32 patients, 11 of glioblastomas, 6 of diffuse astrocytomas, 8 cases of oligodendroglioma, 3 cases of anaplastic astrocytoma, 3 cases of anaplastic oligodendroglioma, and 1 case of pilocytic astrocytoma. In this table, we are showing you the 11 cases of glioblastomas with the IDH status and the overall survival status. So we saw that out of these 11 cases of glioblastoma, 7 cases were IDH mutant, and out of them, 6 were still alive at 16 months and 1 had expired. Out of the other 4 cases which were negative for IDH, 3 were expired and 1 was still alive. The rest of the cases, like diffuse astrocytoma, we had a follow-up of 6 out of 9 cases, and 5 cases were positive for IDH, 1 case was negative, but all 6 were still alive. Oligodendroglioma, out of 10, there were 8 for follow-up, and all 8 were still alive. 7 of them were IDH positive. Anaplastic astrocytoma, we had a follow-up of 3 out of 5 cases, and all 3 were IDH mutant positive, out of which 2 were still alive. In anaplastic oligodendroglioma, we had a follow-up of 3 out of 5 cases, out of which 2 were IDH positive and 3 were still alive. So altogether, there were 24 IDH mutant cases in the follow-up, out of which 22, that is 92%, were still alive with a median survival of 21.5 months. 8 cases were IDH negative cases. They were there for follow-up. And 62%, that is 5, were still alive with a median survival of 15.8 months. So this is the histology showing you the diffuse astrocytoma here. And the IDH positivity, you can see cytoplasmic positivity with perinuclear and weak nuclear staining. So this is interpreted as positive. This is Atrx, in which you can see that the nucleus has not taken up the stain. So this means that Atrx is lost. And we have to look for the internal control. In this picture, you can appreciate that the internal control, such as endothelial cells, have taken up the stain. Then the P53 is showing nice nuclear positivity. When all the nuclei are showing positivity, we give it as a 3+. So the integrated diagnosis would be diffuse astrocytoma, WHO grade 2, IDH mutant, Atrx loss, and P53 positivity. The next case is an oligodendroglioma, which shows again IDH mutant, and here the Atrx is retained. All the cases had nuclear positivity for Atrx, and P53 was 0, that is negative stain. So the integrated diagnosis is oligodendroglioma, WHO grade 2, IDH mutant, Atrx retained, and P53 negative. The third case is of a glioblastoma. We can see IDH mutant is positive, Atrx is loss with the internal control of endothelial cells and lymphocytes, and P53 is strongly nuclear positive. This is a 3+. The integrated diagnosis is glioblastoma, WHO grade 4, IDH mutant, Atrx loss, and P53 positive. So we compared our study with the other studies, and we did not find much of a difference, except that in the median age group, Rasmussen found a much higher median age group to ours, which was our median age group, mean age group was 32. The sex ratio, it was more common in males, and symptoms and location, the most common location was frontal. The other variables which were studied was IDH1 positivity. And in this case, we just saw that some Jankalek had done correlating the IDH mutations using immunohistochemistry and compared it with direct DNA sequencing in 15 cases of low-grade gliomas. And he found that 2 tumors that had IDH mutations were false negative in immunohistochemistry. So that reflects the false negative rate of immunohistochemistry. However, Kappa et al. In a comparative study between immunohistochemistry and genetic testing found that immunohistochemistry was 100% sensitive and specific in detecting IDH1R132H mutations. The association of IDH1 mutation, there was the main association which was statistically significant. It is with the WHO grading. And Atrx mutation, all oligodendrogliomas were non-mutated, and the main association is with the histologic grading again, which is statistically significant. So Tandon J stated that P53 mutation occurs early in glioma genesis and are essentially mutually exclusive with 1p19q co-deletion. Komori et al. Found that Atrx loss in gliomas was equivalent to 1p19q non-deletion, and a combined immunohistochemistry for IDH1, P53, and Atrx improves the molecular classification of diffuse gliomas in daily practice. We compared the combined IHC status of IDH1, Atrx, and P53 against using only IDH1 and Atrx with the WHO grading of gliomas, and both were found to be statistically significant with a p-value of 0.02 and 0.01 respectively. So coming back to the question which I had asked in the beginning, is the study we had done still relevant in the WHO 2021 molecular era classification? So we looked up this paper, which is the latest paper by Komori et al. And here he has summarized that IDH mutation alters the IDH enzymes. It produces a 2-hydroxyglutarate, which is an oncometabolite, which represses the DNA demethylation, leading to genome-wide DNA hypermethylation. And many studies have proven that survival is longer in IDH mutant than in IDH wild type. So in our study also, we showed that IDH mutant cases had a longer survival compared to the IDH negative cases. And in the WHO 2021 classification, it has stated that it is not required to do 1p19q co-deletion by fish once the loss of Atrx and a typical astrocytic histology are confirmed. And Atrx is now used as a surrogate immunohistochemistry marker for 1p19q co-deletion. Regarding the WHO grading, yes, the new classifications had come up because the histological appearance do not always reflect the biologic behavior of each tumor. WHO adopted the combination of CDKN2A*B homozygous deletion along with the histological criteria of anaplasia, mitosis, microvascular proliferation, and necrosis. If CDKN2A*B is positive, glioblastoma is diagnosed irrespective of a low-grade histology. However, Berzero et al. Examined 517 grade 2 gliomas, and he found 29 cases of IDH wild type having a low-grade histology, but with molecular features of glioblastoma, had an overall survival which is longer than that of glioblastoma. So histologic features are still helpful, and molecular features alone cannot predict clinical behavior. Therefore, an integrated diagnosis combining all available complementary information, starting from the clinical to the radiologic, to the histology, to the immunohistochemistry, and the molecular markers is necessary to give a proper diagnosis which will predict the outcome and prognosticate the patient. So we are limited by the less sample size, especially in each category. We initially thought we were limited because of the fish studies, but now with the WHO 2021 telling that fish is not required, so we don't think that is a limitation anymore. But yes, we could not confirm the IDH negative cases, whether they were wild type, because of the unavailability of next generation sequencing. So in conclusion, the immunohistochemistry status of 3 markers versus 2 markers were equally relevant for diagnosis. So for routine glioma diagnosis, a combination of IDH1 and Atrx are sufficient. However, use of P53 is recommended for further prognostication. And majority of the glioma expressing IDH1 mutation are associated with improved survival. These are my references, and this is the latest article, which is very good, from Takashi Komori. Thank you so much.